So welcome to today's discussion video. Today I'm going to be going over mechanisms of cellular signaling and particularly highlighting the example of the MAPK pathway as an example. And then I'm going to go over some problem roulette questions on this subject to kind of get you guys ready for how this will be tested on the exam and kind of like link the discussion material to the exam material, which will hopefully make studying for the final exam a little bit less intimidating. So first, I just wanted to talk about the importance of signaling. So we just got done with talking about gene regulation in prokaryotes and eukaryotes. And signaling is another way that the cell can regulate its actions based on a condition or situation or response. And this includes actually altered gene expression, so similar to like what we were talking about before. But it can also involve other things, like we can see here, it can involve altered metabolism or altered shape, and generally remember shape equals function. Altered movement, so like motility. So those can be altered as well as gene expression. All of these changes can happen in response to either like an external signal or signaling from nearby cells in an organ, for example. So essentially what we need is a way for the cell to be able to translate that signal into something that changes its function so that it can adapt to the condition that the signal is trying to warn the cell about. And how we do this is with effector proteins. So we can see that each of these changes that I was talking about is regulated by an effector protein, something that will cause that change to happen. And we want those conditions to be available under certain circumstances. We want that to be able to be turned on. And the whole rest of this lecture, or the rest of this discussion, is going to be talking about how we can get those effector proteins to be activated based on receiving a signal. So where we've seen this type of mechanism before, hopefully you can think of a lot of examples, but just a couple are the presence of lactose or tryptophan would affect whether or not those genes, the operon controls, would be transcribed. Emergent properties, so we can think of examples where an individual cell won't have like certain genes being expressed, but suddenly when there's like a biofilm or a big mass of cells all together that are signaling to each other and coordinating with each other, you can get this emergent property emerging. And a lot of other examples that we can hopefully think of, because essentially we're just relying on this whole structure equals function thing, that once we're able to induce some sort of change, we're going to see a change in the cellular response. Okay, so the two key assumptions in cell signaling is that one, the cell needs to be able to receive the signal in the first place. And then once the signal is received, the cell needs to be able to change somehow in response to that in a way that will affect its function. So if either one of these aren't met, or both of them aren't met, you're not going to be able to change in response to a stimulus. So if you think about an example of, if you can see this woman here is trying to call the fire department, presumably, if her phone is dead, or if no one's picking up at the fire department, or something like that, if the signal is not able to be received, then obviously that's going to prevent a solution to the problem, which is the fire truck coming and putting out the fire. But let's say she gets through to the firefighter, but he speaks a different language, or he doesn't have access to a fire truck, or something like that. If the signal's received, if it doesn't actually produce the change that you want ultimately, then it's not really going to be a mechanism of like regulation or like cell signaling. Okay, so when we say signal, we're actually referring to a lot of potential things. Signals can be proteins, carbohydrates, steroids, pathogens, amino acids. It can be basically anything. So that's why we call it just a signal, unless we're talking about a specific pathway, in which case we know what molecule we're talking about. But yeah, because signals can be a wide variety of things, we always just kind of lump it under that nebulous term unless we are like saying something about a specific pathway. There are two main ways that a signal can bind to the receptors. One is intracellularly. So if we have a signal ligand that's able to cross this membrane on its own, and it's able to bind to an intracellular receptor, then this intracellular receptor can change its shape while it's already in the cytoplasm, and then cause some sort of conformational change that will change the function overall in a way that we care about. However, if the ligand can't cross the membrane on its own, then obviously intracellular receptors would do us no good because you would never be able to reach that. So in this case, you're going to have these receptor proteins that span the membrane so that we can bind on the extracellular side. And that binding is going to cause some sort of change on the cytoplasmic side which will induce the rest of the pathway that's needed to make that final change that we care about. So we can try to predict which of these binding types is more common. And actually, so the cell surface receptor kind is going to be more common. It's going to be more rare for a substrate to be able to cross the membrane on its own. So generally, this is going to be a little bit more common, but we can think of examples of intracellular receptors. And steroids is probably a big example of this. Steroids are able to cross the membrane, and they can bind and go into the nucleus. And I'll show you more about that later. But that's a good example of an intracellular type. But hopefully you can remember this was from the lectures in exam 1. But in order for something to cross the membrane on its own, it's going to have to be like really small and nonpolar and uncharged and hydrophobic and like all this stuff. And anything else is not going to be able to cross. So anything that's like on the more hydrophilic, larger, polar side of things is going to require those external receptors. Okay, also just an additional note. So the same ligands can elicit different responses. So a good example of this is actually with adrenaline. So we can think about its effect in cardiac cells versus intestinal cells. In cardiac muscle cells, adrenaline is going to cause an increase in contraction, whereas in the intestine, adrenaline is going to lead a decrease in contraction. So we can probably predict, well, there must be a lot of similarities between these two pathways. There must be similar receptors because they're both binding the same molecule, so they must have similar shapes in that way. And they're both leading to something that has to do with contraction. We can think like we probably have a lot of the same components just working in opposite directions of each other. So like either enhancing binding or preventing binding, et cetera. Having secondary messengers and like intermediate steps that are working in different directions from each other. And hopefully this should make sense on a broader scale because we can think about like why did the human body evolve these two different responses to like the same thing. So if you think about the nervous system and our fight or flight response, things like that, we want to be able to divert resources to the things that are going to be like most useful in that circumstance. So we're gonna, in this case, if we have a high adrenaline situation, we're gonna divert a lot of our resources to our cardiac system. We want to be able to provide oxygen via our heart. So that's gonna be really important, but digestion is not gonna be high on the agenda. So that's why we can kind of divert the resources to the heart rather than digestion in that case, because we're presumably in a circumstance where like running and fighting is going to be really important. Whereas under normal conditions, you're not as worried about that, and you can just as easily focus your resources on digestion as well. And similarly, we can also have different ligands that elicit similar responses in the same pathway. So it's kind of like the converse of this first bullet point. So typically this will occur with signal analogs. So analogs are essentially just like really similarly shaped molecules. And we can think like, okay, if the binding of the signal to the receptor is based on shape, if you have an analog, you're going to probably get a similar type of binding because you have that similar shape that will allow it to bind in the first place. It might differ in their affinities. So you can have an analog that's like less effective, but overall you're gonna assume that they're still gonna be able to activate the same pathway. And this is actually the crux of like pharmacology in a lot of cases, being able to use a drug as a way to like stimulate a process that's not occurring naturally. And you're able to do that by like making something that looks really close to a ligand that would normally occur. And if it's not, then you can just add that. You can just add that and hope that you're gonna get the same effect in the end. Okay, so one of the main ways of activating this whole system that I want to talk about is G proteins. So G proteins refer to this peripheral membrane protein that we see here. And G proteins are always going to have this, in its default state, it's going to be bound to this GDP molecule, and it's going to be inactive in this state. So always remember that if it's bound to GDP, it's going to be just chilling, not doing anything. And in order for this to be able to do anything that will allow the pathway to occur, it's going to have to be activated. And the activation is always going to happen by having the G protein being bound to GTP instead of GDP. So we can think of GTP will always be active, GDP will always be inactive in this case. And how this relates to what we were just talking about with signals, is that we can have these receptors that require a signal binding to a receptor in order for some sort of conformational change to happen that will allow this GTP to displace the GPP from here. And so this can actually be seen a little bit more clearly in this next picture. So we can see this ligand is binding, and then that's causing like this end shape of that receptor to change. And then we don't really have to know the specifics of the details of this part, but like something about that interaction right here is going to make this GTP binding more favorable. So it's going to let the GTP come in and bind, and the GDP will be displaced. And then now, since that is in its active form, this G protein is now active, then it can proceed to the next step in the pathway and bind to the effector protein, which will produce all the secondary messengers and intermediates and stuff that will take us to the net effect, like the effect that we really care about. Okay, so another really important thing to talk about is receptor kinases. So when you see kinase, you can hopefully think to yourself, oh, that indicates enzyme because it ends in ase. And so receptor kinases are enzymes, but they're also receptors for signal molecules. So the one that we saw before, the one that we were just looking at in this example here, wasn't really an enzyme because essentially we were just changing its shape, which allowed something else to happen just because of like how it was positioned. But it wasn't really like catalyzing a reaction on its own. Whereas with here, we're gonna see that we're actually catalyzing a reaction. And so receptor kinases are gonna have these like two separated subunits when they're in this unbound inactivated form. And when they bind the signal molecules, what's gonna happen is a phenomenon called dimerization. And that sounds confusing, but essentially that's just gonna cause those two units to come together and form a single complex. And so when this complex is formed, this is the part where we get the enzyme activity occurring. So this is going to allow phosphorylation to happen. It's going to hydrolyze ATP. So ATP is going to be broken down into ADP. And so we'll have that extra phosphate group from that reaction. And then that extra phosphate group is donated to what's called a phosphoprotein. And here, in this case, the phosphoprotein is what leads to the rest of the pathway. So it's kind of equivalent to the G protein from the previous example that we just saw. And again, just like remembering structure equals function, we can assume that something about this phosphorylation changes the behavior of the phosphoprotein, which allows that next step of the path to be activated, and so on and so on. Okay, so we can kind of compare G proteins versus phosphoproteins a little bit. So both of them are, in a way, activated by the addition of a phosphate and deactivated by the removal of a phosphate, just in different ways. So the G proteins were activated by a non-covalently bound GTP molecule as opposed to GDP. So that still has one extra phosphate group, but it's the whole GTP molecule that's bound. In phosphoproteins, we have a covalently bound phosphate group that was donated from the hydrolysis reaction of receptor kinase. So there, we're just having that extra phosphate group, not the entire ATP molecule. So that's like a primary difference. And also, so the G proteins will be deactivated when that GTP gets hydrolyzed back into GDP. Whereas with phosphoproteins, they require a phosphatase enzyme, which is going to cleave off that phosphate group that we added, and it's going to cause that to become inactive again. So we can see this here in this diagram. So here's the phosphoprotein, and here's the G protein. And so we can see in this case, so we have GDP exchanged for GTP, and the whole GTP molecule is being bound. And so this is being an active, so this is an active form, allowing the signal to be passed on. Here, what's actually happening is ATP is being broken down into ADP, which kind of seems like it's moving in the opposite direction here, but we have to remember that what we're doing is we're donating that phosphate group from that reaction to this phosphoprotein. So ultimately, we're still having this net effect of like, we have an additional phosphate here, an additional phosphate here. And then in both cases, to inactivate the two, we have to get rid of that extra phosphate that was added. Okay, and also just another note, so sometimes you'll see receptor kinases, and sometimes you'll just see kinase, and sometimes you'll see protein kinase. So I wanted to clarify. So kinase just refers generally to an enzyme that will add a phosphate group to another like target molecule. This is generally catalyzed by ATP hydrolysis, or it can be other high energy molecules in some cases. And the 3-phosphate group is donated to the target molecule, which allows it to be activated. So sometimes those enzymes are receptors of the signal molecule, which we saw just recently in the slide here. It's a receptor, but it's also a kinase because it's able to catalyze this reaction. But we can also have kinase activity acting later on in the pathway that's not actually receiving a signal, but using phosphorylation as a method for some other change that we care about. So in that case, you'll just see that referred to as kinase or protein kinase. Those are equivalent terms. So just remember that receptor kinases are kind of just like a subgroup of kinases that refer specifically to the ones that are binding the signal as well as causing that phosphorylation of the phosphoprotein. Okay, so now moving on to kind of like the rest of the pathway. Second messengers are hugely, hugely important when talking about signal transduction and cell signaling in general. Second messengers are essentially any small non-protein signaling molecule. So it can, again, kind of like signal, it can be, like kind of like the term signal, it can be nebulous because it can be referring to a wide variety of things. But the key thing to remember here is that it's a non-protein. And generally they're able to diffuse easily through the cell. And they can trigger like a wide variety of things depending on what second messenger you're talking about. And so the reason why this is important is, one, diffusion is going to be really important for signal amplification, so that the second messengers can reach a lot of whatever it's doing in the next step. If it's activating an enzyme, it can reach those enzymes, etc. And the ability for different second messengers to trigger different things is really important because we want to be able to respond to a wide variety of situations and conditions. And so having that flexibility kind of allows us to like tailor our responses to signals a little bit more. And then signal amplification is just referring to this mechanism of how at each step of the pathway, we're like getting progressively more and more of a response. Because you can see here, we start off with one signal binding to one receptor, which can activate multiple G proteins. Then each of these G proteins are able to produce a lot of the CAMP, and you have these multiple activated G proteins all creating new CAMP. All the new CAMP is able to activate a whole bunch of protein kinase A. So at each step, we're getting progressively more and more activation. And why this is really important is that we're able to generate a large scale response to compared to the signal received. And this can be really important because like if you're like a cell that's located near poison or something, you don't want to have to require a ton of poison being present in order to like transcribe those efflux pumps or something to get it out. You want a little bit of it to be able to produce a large effect. And this amplification process is going to allow that. Okay, so now I want to talk about the MAPK pathway in general. This pathway, it kind of combines the elements of receptor kinases and G protein. The ultimate goal of MAPK is to initiate cell division. And how that works, the first step is that it wants to take this mitogen signal, and hopefully you can kind of predict, based on what I just told you about what MAPK is trying to do, about what mitogen means. So gen as a like suffix here refers to like creation. So like we can think gen, genesis, creation. And then mito is mitosis. So it's essentially creation of mitosis or initiation of mitosis more specifically. And then that ultimately we want to result in a net change of upregulating the transcription of genes that are able to induce mitosis. And so the MAPK pathway is how we get from that signal to that effect. So the first step in that process has to be binding. And we're told that mitogens are polar. So we know that they're not going to be able to utilize intracellular receptors because they're not going to be able to cross that membrane on their own. And sure enough, we can see that in this graphic. You see, hopefully you can tell that this is going to be a receptor kinase because we have these two units that are going to dimerize and catalyze a reaction, which we'll talk about. But essentially, so this signaling molecule is going to bind, causing this activation to occur, formed. And what will happen is this dimer will bring a RAS activating protein to the G protein and will cause the GDP that's being formed to be phosphorylated. So we're donating a phosphate group to this, we're not displacing the whole GDP molecule with another GTP molecule. So like if we looked up here in this example, essentially we were using that conformational change to displace GDP with GTP that we could see here. Whereas now we know that this is a kinase because it's able to phosphorylate this directly. And now since we have this GTP being bound, we know that the G protein here is going to be in its active state, which is going to allow it to kind of continue down, down the pathway. Okay, so the next step in the process, you can see we have this now activated G protein. What's going to happen is it's going to interact directly with a MAPK protein, which currently is inactive. But when it directly binds and interacts with the G protein, that's going to change this to its active state. And again, you don't have to know what specific like conformational changes are happening to allow that to happen, but we just know that that interaction is going to allow this to be activated. And then we can see from this activated MAPK, what's happening now is we're getting this ATP hydrolysis reaction that's donating that extra phosphate group to an inactivated MAPK, which again allows that to become activated. And now, again, we're getting this hydrolysis reaction, which is going to donate an extra phosphate group to an inactive MAPK, which is going to activate it. And then once this step here is activated, it's going to be able to enter the nucleus and be able to bind transcription factors and ultimately allow the transcription of those genes involved in mitosis to occur. So ultimately, we can see how we went from just this signal molecule, this mitogen being bound, all the way through how the actual gene expression is being regulated. All right, so now here you can just see I summarized this whole pathway into a quick little blurb. And if you are ever like confused about what's happening here, a good thing to do is remember what these proteins stand for. So if I told you MAPK stands for mitogen activated protein kinase, and then MAPK is just like a MAPK kinase and a MAPK kinase for all these proteins, that should kind of tell you what their role is in the pathway itself. So we could see here, this was our G protein that's activated, but it's not a kinase. So what's happening here is not a hydrolysis reaction. We're not phosphorylating this. We're having like a direct interaction, a binding between these two, which is activating this. So remember that when we don't have a kinase, we're not doing this phosphorylation. But at each of these steps, when we have this MAPK, the action that it's taking is it's donating that phosphate group via hydrolysis to activate the next protein. And then this, which is also a kinase, is doing the same thing. It's donating that extra phosphate group to another protein. And then here, it doesn't really like show specifically, but the way that this is able to bind to these transcription regulators is by phosphorylating them. So that is still acting as a kinase by phosphorylating the next step in the chain. So that should kind of tell you, okay, where do we have phosphorylation activity versus just like direct binding activity? When you have a kinase, that's going to tell you we're phosphorylating. Okay, so I wanted to kind of go over some problem roulette questions real quick and talk about modifying pathways and comparing like upstream versus downstream and stuff for modifications. This concept will be really useful in answering questions about the effect of a certain like drug or inhibitor or a mutation at a certain point in the pathway. It'll tell you like what you can expect and allow you to make predictions based on that. So this interesting problem roulette question states, a human cell line has a mutation in the MAPK gene that alters the MAPK protein, making it constitutively active. The mutant line exhibits overproliferation of the cells. Which of the following treatments would reverse this overproliferation phenotype? And then you can look through all the options on your own. But, okay, so given that question, you can look at the transduction cascade here, and we can say, okay, the mutation that it's talking about says that this MAPK protein is always going to be active, which means that this has been upregulated. So we're constantly getting these mitosis initiators being generated, which is what's causing that proliferation of cells. That's going to be pretty damaging because you can cause tumors that way, or you can even have organ enlargement. There's been some interesting studies that show really dramatic organ enlargement in people that have non-functional MAPK pathways. Because of this, it's not able to regulate this being turned off. But so back to the question, if this is always going to be active, what change can we make that will still allow this to be stopped? Hopefully it should be clear that if we already have active MAPK, it doesn't really matter what's happening before it, because all these steps are trying to do is to get to this activation step. That's the whole like function of those previous steps. So if you have a shortcut, essentially a mutation, that's saying we don't even need those extra steps to activate it, it's always going to be activated, you can conceptualize the transduction pathway as starting here. Because now we're always going to be able to continue down the path since we're already fulfilling this requirement of activating that MAPK, so you can continue down the rest of the chain. So in order to stop that, we have to modify something that comes later in the chain. So we can call that downstream. And by doing that, we're able to block the pathway, the rest of the pathway from occurring, even if we have a previous step that's always going to be on. So the answer to this question was adding a MAPK inhibitor. And hopefully that should be clear given the structure, because we can see, okay, if this is always going to be on, if we have an inhibitor that will prevent this from being activated, then having this always be on doesn't really do much for us, because the whole point of this activation step was to activate the MAPK. You're saying, okay, now the signal pathway is starting here and we're inhibiting it. So now we can say that the total effect, the transcription of the mitosis initiating genes are going to be blocked because of this. So hopefully that is clear. Another interesting one, GTP gamma S is a GTP analog. So remember what I said about analogs, that functions like GTP, except that the terminal phosphate cannot be cleaved off. If you added GTP gamma S and the signal to cells containing the MAPK signaling components, so it has the pathway, which of the following would not be an expected effect? So first I want you guys to remember, okay, GTP gamma S works like GTP, but we can't have that terminal phosphate being cleaved, so we're never going to get the situation where we had this GTP going back to its GDP form. So functionally what that means is that a G protein with this GTP that's added is always going to be, or like with this GTP analog, the GTP gamma S is always going to be in its active state. It's not going to be able to be turned off. And so we can use that information to hopefully answer this question. A good way of approaching this is saying, okay, where in the pathway are these functioning? And then looking downstream of that to see, okay, what are we blocking or activating? Like what's going to be the effect based on what's downstream of these two things. So looking back up here, we say, okay, if this is going to be an analog that's going to be bound, we know that this RAS protein is always going to be activated, because we're never going to be able to return to this inactive form. And once we have that activated form, we're always going to get the rest of the pathway occurring, assuming that we don't have an inhibitor or a mutation or something, which we don't in this case. So anytime that you have that analog, you're always going to get this whole rest of the pathway being expressed. And so how we can answer that is, A, none of the options are correct because all of the following are expected. And this is because the question asks what would not be expected. And we actually expect every single one of these. So hopefully you can look at these and say, okay, MAPK would be continuously active, MAPKK would be activated, cells would be able to activate MAPK, RAS would be constitutively active. And we know that these all have to be true because we're always going to have that G protein in its active form, which is kind of like the big kahuna of controlling the rest of the pathway. All right, and now finally, very similar question, but might be kind of tricky. GTP gamma S is a non-hydrolyzable form of GTP. So again, it can't cleave off that third phosphate group. So which of the following treatments would lead cells to, or which of the following treatments would lead to cells with continuous proliferation? So we're saying which one of these options would not block the pathway from occurring? Or inhibit MAPKK, MAPKK, MAPK, or receptor kinase. Back to the previous question we just answered, we are going to have this GTP analog that's going to be constantly bound and activating this, this RAS protein. Now, remember that any modification that occurs upstream of the thing that you're talking about is not going to affect the pathway. Anything that's upstream is ultimately just trying to get to the state for the next thing in line to be able to do its part, to be able to be active for the further steps. So when we're blocking the activity of this receptor, it doesn't really matter in this case because the whole point of the receptor was to activate the RAS protein. But we're being told in the question that this is always being activated anyway. We are not going to be able to ever have it in the inactive form. So taking away this receptor doesn't affect the end result because we're still going to always have this form. And once we always have this form, we're always going to get all of the following steps happening. So when we look back at the actual question, we can see that, so it says, so when we look back at the question, which of the following treatments would lead to cells with continuous proliferation? We know that it has to be E because upstream of this analog that's activating the G protein is not going to have an effect. Whereas each one of these inhibitors, the ones of the downstream kinases, those are going to stop the transcription of those genes from happening, which is going to cause the proliferation to stop. So that's why this answer E is the only correct choice here. Okay, one last question. Which of the following is not true of the MAPK signaling pathway? And I'm gonna let you guys kind of like read these on your own. Ultimately, I just wanted to put this one in here to highlight or like reiterate the fact that ROS is different than the rest of these steps, all the MAPK kinases. All the MAPK kinases, hopefully the name will help remind you that how it's working is it's phosphorylating like the next thing in line to activate it. But step 3 is different because this RAS protein is not a kinase. So that initial MAPK protein is becoming activated via a different method. Here it's going to be binding directly to this RAS protein. All right, so I just wanted to plug some special examples really quick. These both are like essentially the same fundamental processes and mechanisms that we've just talked about, but under slightly different conditions. One of them I brought up previously, which was the steroid hormone receptors. And the reason why this process is unique is, well, one, it has these intracellular receptors. So we know that the steroid hormone must be able to cross this membrane by itself, and it does. But what's really unique is that this whole complex, this receptor and steroid binding complex, is actually the effector molecule. So this whole thing is going to be translocated into the nucleus, and it's going to bind to an enhancer sequence and alter gene transcription just by itself. So like typically in other pathways, we would see that this binding would induce a change of something else, which would induce something else, which would induce something else, so on. But here, it's just this complex directly that is actually causing the change that we care about. So that's why this one is kind of interesting. For the ligand scenario, this one's interesting because the signal is essentially just acting as a gatekeeper for a channel protein, which generally just allows solutes to be moved in and out of the cell. So this one is a little bit different because the change that it's enacting is also not like, it's like different than the receptor kinase mechanisms and stuff, because now we're just regulating solute. So ultimately, if you're to remember just a single thing from my discussion today, it's that structure equals function. So the entire crux of how signaling works is that we're inducing some sort of conformational or structural change in a path that's usually going to be a multi-step pathway that's going to allow the signal binding all the way down to the step where we're getting that change in function or behavior that's going to allow us to adapt to that original condition that the signal was like telling us about. So yeah, it's all about structure and function. Now I wanted to go over some quick things for the discussion exercise and quiz. I would highly recommend working on the exercise before the quiz this week. If you look at the first quiz question, you'll be like, what is this even talking about? And that's because it's referring to a specific pathway that's in the exercise itself and wasn't talked about right here. So you're not going to know what that is until you do the exercise. And one piece of advice that I have for you is trying not to get too bogged down about like all the nitty-gritty details of like understanding every single word and like understanding every step of the whole pathway at a time. etc., just because it can be kind of overwhelming. I would say, like, when you're trying to answer a question, look at the step that it's talking about, look at what's directly preceding it and what's directly following it to see, like, where is it oriented in this pathway? What's upstream? What's downstream? Like, really localize to the question, or to the part of the pathway that you're trying to figure out, so what's going to happen. And then this will be really useful, just like focusing on what part of the pathway you need to be looking at for a specific question. And also just to kind of go over how to analyze these tables. So you'll see that you're given a pathway and then these like results. And so treatment, what this means is that essentially this treatment column is just saying, okay, these are the things that we're trying to analyze the effect of. The inhibitor column, minus just means that the inhibitor is absent, plus means the inhibitor is present. And so you can use the difference between the presence and absence of an inhibitor to kind of see, okay, what effect does the treatment have on the overall gene expression? That's going to be really important. And then when you're looking at the target gene expression, just knowing that these are arbitrary units. So we're just comparing relative to each other. So again, it's like really helpful to compare, like, okay, look, when we added the forskolin inhibitor, we went from 25 to 5. That's a really huge jump considering the next step, adding that inhibitor only decreased by 1. So like that can kind of help you isolate like what is happening in the pathway, like where is the like drug or mutation or whatever you're trying to identify, like where is that targeting in the pathway. And also just some other piece of advice. So exam 3 is in 9 days. So it's coming up really quick, and I want you guys to be able to focus on how like this material is going to relate to the exam questions. So what I would really recommend that you do is work on the cell signaling problem roulette questions tonight while it's still fresh and you've like just learned it and you're ready to go. Taking note of anything that you don't understand, any questions that you got wrong, anything that you're confused on, whatever. And then you can either ask on Piazza or just like try to study with your notes and try to figure out the answer yourself. Or if it's like really troubling you, you can talk to either me or Lori or Sarah in office hours, etc. Really like addressing those problems right away before the exam comes up even closer. Also, another like really helpful piece of advice I think would be like when you're doing these problems to not just be able to identify, okay, this one is clearly the right answer, but be able to actively eliminate the other answers. Say like, why would this one not be right? Why would this one not be right? Why would this one not be right? Because by doing that, you're gonna be better adapted to situations where like on the exam, we might flip the situation where we would say like, what is not true about this versus what is true about this? Things like that. So thinking through that exercise is really gonna solidify the concepts in your brain, so you'll be more flexible to any exam question that's thrown at you. Yeah, and so I would start working on all of that, and then as the exam gets closer, just like redoing those cell signaling problems that you were initially stuck on or got wrong. And if you're able to do those like easy peasy after you've like done this extra review, that's great. If you're still having trouble, then that's going to be an indication that you need to focus more time on like that specific segment, or like contact a GSI or professor so that we can like help clarify that before the exam. All right, so this is all that I have for you guys. Again, try to work on the discussion exercise before the quiz, and if there are any questions, please let me know, because I know that this can be kind of confusing for the first time that we're looking at it. So, all right, thank you guys.